Now we would like to welcome on stage as chairs Dr. Loisa Sloizo and Assistant Professor Dr. Vasiliki Danilatu.
It is a great honor for me to present Professor Arne Bogart. He's full professor of pediatric oncology and hematology. Became the head of Department of Pediatric Oncology and Hematology at the Ludwig Maximilian University Hospital in Munich. Since 2006, he's the head of the Department of Pediatric Oncology, Hematology, and Clinical Immunology at the Heinrich Heine University in Düsseldorf. His research interests relate mainly to leukemiogenesis, bone marrow failure in children, bone marrow transplantation, role of fusion genes in leukemiogenesis, diagnostics, chromosomal aberrations, immunodeficiencies, function of microRNAs, and their role in the malignant transformation of hematopoietic cells. He published and co-authored more than 350 peer-reviewed papers, and his research group discovered a novel inherited T-cell Hodgkin's lymphoma. Professor Borchardt served as a member of the board of the European Hematology Association and as well as the scientific advisory board of various funding agencies. Also, I want to say that Professor Borchardt is a big friend of Cyprus. He has contributed greatly to the management of our patients. He since many years collaborating for the clinical management, and he's always also member of the research team that we established in Cyprus for childhood cancer. Professor Borchardt will talk to us about prevention of childhood acute leukemia, fiction or future reality.
Thank you so much, Loisa. I mean, now we spent nearly half of my talking time, but no, it's a great pleasure for me to be here. Also, thanks to Professor Patricios for having me here, and we had this wonderful night yesterday. So, as Dr. Loisos told you, I'm a pediatric oncologist, and it's a very narrow and a very narrow field, I would say. So the challenge, that brings the challenge that we have to present something new in terms of scientific content. On the other hand, it's a very specialized field, so I didn't want to talk about my favorite gene or favorite pathways and to go too much into detail, because such details usually will be forgotten soon. So, and I thought it would be better to present more or less a vision or an idea how we could probably in future prevent the most common childhood malignancy in childhood. So, and when you see this boy, of course, he looks very unhappy, and that's what we usually see in the clinic. And of course, the disease starts in the bone marrow and infiltrates several organs, several extramedullary organs like the brain and so on and so forth. And the next couple of minutes, I will talk a little bit about the complex interplay between genetic predisposition and mainly infection. So we think that childhood acute lymphoblastic leukemia, and to be more specific, the B-cell lymphoblastic leukemia, derives from such a complex interplay. So what is my talk about? I will briefly summarize what we know about with the theories, how childhood ALL develops, and I will spend a few words about the link between infection and childhood ALL, a few words about genetic predisposition. We heard this term a couple of times before, and then some studies from our lab, summarizing some animal studies to try to understand better what happens when you expose a genetically susceptible animal to an infectious challenge, to an infectious environment. And then, of course, somehow a dream or a vision, could we probably prevent the disease in future? But that's maybe something for the next generation of pediatric oncologists. So the idea that infection is somehow linked to the development of childhood leukemia is by far not new. So here you see a quote from colleagues from England almost 100 years ago, and it's really remarkable what they wrote at that time. We incline to our evidence to the belief that the solution of problem of leukemia lies rather in some peculiar reaction to infection than the existence of a specific agent. So the idea is not that there's a leukemia-inducing virus as such. There are some such examples, mainly in adults, but we rather think that's a very strange and abnormal reaction of the immune system to a more or less harmless infection. That's what the current belief is. And just to drop a few names, that's the first one who recognized this age peak, as you can see here, this huge age peak in ALL. Usually the kids come into the clinic between, let's say, 2 and 6 years of age, and this age peak is very peculiar, very special for ALL, and we don't see the same age peak in other pediatric malignancies. And so Ward, by the way, he was a military doctor in the First World War. He collected a huge number of cases and for the first time recognized these age peaks. And then later, a couple of colleagues, Leo Kinlan from Oxford and Mel Greaves from London, they somehow refined the theory how infection is linked to childhood ALL. So, and just to summarize the work of Mel Greaves, a close colleague and to some extent friend of mine, he was the first who brought up this two-step model that the first genetic hit occurs in utero during fetal development, and then a so-called preleukemic clone is formed. This preleukemic clone can be either inherited by genetic predisposition or in most cases can be somatically acquired during fetal hematopoiesis. And that's, as you can see in a couple of minutes, occurs quite often. It's not a rare event. Many healthy newborns are born with such a preleukemic or at least clone or at least a cell clone with a certain amount of risk to become a full-blown leukemia. But then something else should happen after birth. Some, I would say, abnormal or dysregulated immune response that really drives the outgrowth of the preleukemic clone. So, and that's a two-stage process. First one before birth, and the second after birth. And there are a number of factors and genes involved there. I think that's probably not of general interest. But there are a number also of epidemiological and observational evidence for this hypothesis. And one of those is presented here. For instance, a couple of years ago, there was an outbreak, if you wish, long before COVID, an outbreak of childhood acute lymphoblastic leukemia in a school in Milan. And they had 7 cases diagnosed within 4 weeks with acute lymphoblastic leukemia. And you can imagine it caused a lot of media attraction, and the school was closed, and the parents became very concerned about the school. And it was simply by accident, so to say, but many children were diagnosed with a flu, with a swine flu, prior to this outbreak of ALL, meaning that was an abnormal response to a more or less common viral infection. So we know a couple of those so-called leukemia clusters. That's not the only one in Milano, but maybe the most recent one. So it happened from time to time. So a few words about the inherited genetics of ALL. I don't want to go into much detail, but we have to distinguish between so-called low penetrance susceptibility genes, so genes which are, or risk alleles, I should say, shared by you and me and many others, but they have a very modest or moderate risk for developing leukemia. And the other risk alleles, they are much stronger, so to say, in terms of their effect, and fortunately, they are much rarer. In very rare cases, we can see familiar acute lymphoblastic leukemia, so affecting, let's say, 2 kids in the same family. And we know, meanwhile, a couple of genes here. You see hematopoietic stem cells differentiated along the B-cell lineage to the mature B-cells, and we know the genes responsible for this genetic predisposition, for this susceptibility to develop a leukemia later on. So that's to some extent inherited from your parents. Much more common is the so-called somatically acquired predisposition. I'm still not talking about leukemia, I'm talking about predispositions, it's a chance to get leukemia later on. And that's much more common. And we and others did studies in which we screened cord blood cells or cord bloods from normal, healthy newborns, and to look at the frequency of such preleukemic clones. And there are a number of studies here. Daniel Hein from my lab did a number of studies in which he looked at different types of genetic predisposition. The most common one is the so-called EDV6-RANX1 fusion gene, and he found it, this preleukemic fusion gene, he found it in up to 5% of all healthy newborns, and some other fusion genes at a much lower frequency. But anyway, the frequency is much higher than the number of kids later on developing leukemia. So the great majority of children clear the preleukemic clone and never develop leukemia later on. So that, of course, brings the question, what's going to happen in those kids who really develop leukemia? And that was a point where we addressed this question by a number of animal studies. Here, just a recap: the normal B-cell development starting from the bone marrow, from hematopoietic stem cells, and then ending up with the peripheral blood, with mature B-cells, memory B-cells, or even plasma cells. And in animals, of course, you have a chance to model this process. You have simply to mimic or to resemble the genetic predisposition. I said we know the genes which are responsible for it. And then we can introduce the so-called first hit by a transgenic mice, and then we can expose those mice to infectious stimuli, to an infectious environment. And that's exactly what we did. Here, a couple of mouse strains with different types of genetic predisposition. And as long as you keep such animals, so to say, clean in clean cages without specific pathogens, they never develop leukemia. But when you start to, you may recognize this funny microbes I put around the animals. So when you start to challenge those predisposed mice with microbes, we simply just transferred them from clean cages to so-called dirty cages and exposed them to a number of stimuli. Then we could observe leukemia development closely resembling what we see in the kids. So in that respect, the models are pretty good, and we published that in a number of papers. For instance, here this PAX5 heterozygosity, one of my favorite genes because it's of tremendous importance in B-cell development. So this PAX5 heterozygotes developed, 20% of all those heterozygotes developed leukemia, but only when they were challenged by infectious stimuli. So, and that brings me to a topic of the microbiome. So you all know, when you look in the mirror, you think it's all human, but I would say only the minority of our cells are really human. So, and when you look in the genes, it becomes even more pronounced. For instance, we have 30 trillion human cells, but we have much more microbial cells in our body, each of us. And when we look in the genes, the difference becomes even more impressive. We have all around about 20,000 genes in our cells, but a huge number of genes coming from the microbiome or from the microbiome. So that means that we do neglect 99% of all our genes in the system. I have spoken about this interplay between genetic predisposition and infectious challenge. So we set up a study. Very briefly, we took mice with a genetic predisposition, the so-called PAX5 heterozygous mice, and we mixed them up in different cages with healthy, normal mice without genetic leukemia predisposition. So we kept them in mixed cages or in cages where only one genotype is housed into. And then we had different facilities, so-called specific pathogen-free facilities, clean facilities, and this dirty facilities. And then we collected stool from the mice for over a period of time and determined by a more or less sophisticated approach the microbiome of those animals and asked the question, what is a microbiome, so to say, a biomarker? Can we see any changes in the microbiome related to leukemia development? And to cut a long story short, that cartoon here somehow summarizes our findings. So when we have a predisposed mice, as I said, a PAX5 heterozygous mice, and these mice, we can see, even the mice is completely healthy, not developing leukemia, but we can see some mild abnormalities in the B cell compartment, meaning that this genetic predisposition does something, but not enough to get the full-blown leukemia. And as I said before, when we challenge those mice by infectious stimuli, then of course we can develop leukemia. But in another set of experiments, we asked the question, what's going to happen when we destroy the normal, healthy microbiome? And we did so by antibiotic treatment. It was a very heavy cocktail over 8 weeks. We really decontaminated the total gut of some animals and couldn't see any microbes during that time. And then, much to our surprise, we could see leukemia development even in those animals when they were kept clean, meaning that the microbiome somehow protects the development of leukemia. When you destroy the microbiome simply by heavy antibiotic treatment, then you can induce leukemia development even if the environmental trigger, so the dirty cage, is missing. So, and that, of course, at least in theory, brings some ideas how you could prevent the leukemia development. And a good thing, of course, would be to maintain and to nurture your normal protective microbiome. And how kids do that, and here it fits, or it meets also several epidemiological studies, and Dr. Erdmann is going to talk about epidemiology. I have no clue about epidemiology, but there are a number of epidemiological studies saying that the risk for leukemia is reduced when you have when you go to kindergarten, to daycare, when you have a lot of siblings, and also studies showing that the close contact to pets and to livestock is reducing the number of acute lymphoblastic leukemia. breastfeeding is a very good thing for several reasons, but also in terms of reducing the risk of getting leukemia and so on and so forth. So a number of epidemiological studies supporting that view that the microbiome somehow is of importance, or a disturbed microbiome is somehow leukemia promoting. So, and we could even think about so-called trained immunity-based vaccines. That's a very fancy term. Some vaccines which are not very specific against one specific agent. So they have a more broader immune stimulatory effect. Nobody knows exactly what's going on the molecular level, but at least we could envision such a procedure, this trained immunity-based vaccine. One of the famous examples, by the way, is BCG, vaccination against tuberculosis. And it's also known that vaccination against tuberculosis is not very effective against TBC, but it reduces a little bit the risk of getting leukemia in children. So coming back to this more or less future vision, so the idea would be that you have very fancy molecular tools, and when you look in the mirror, you can see at least your skin microbiome very quickly, and you can determine all the different kingdoms and species. I'm not an expert in microbiology, but let's assume we can do it very quickly without much lab work. And then we see, of course, a huge number of different bacteria and so on and so forth. And then some computer tools may help us to identify those gaps in our microbiome, or so bacteria which are, so to say, or cause a risk for develop leukemia. And then, of course, it comes to the idea that we can do something for prevention of the disease by adding something which is missing, and that example here, for instance, lactobacillus. So as I said, it's only a future vision. We are not there, but at least we know much better what's going on during the first 1 to 2, 3, 4 years of life, and what's going to be wrong in children who develop B precursor leukemia. And with that, I would like to thank here some of my co-workers, my team in Düsseldorf, and I would especially thank Stefan Janssen, who is our bioinformatician and became recently a professor in another university. So when you are a bioinformatician, you can get a professorship very early on, because such people are really urgently needed everywhere. So they get professorship around age of 30, I should say. And Ute Fischer, who is, so to say, my senior postdoc and runs my lab. And I thank you for your attention. Happy to take questions.
Professor Burkhardt, thank you very much indeed for this great, excellent presentation, and that you gave us new insights about the etiology of leukemia, and hopefully sooner than later prevention. We have time for one question, please. Anybody? No questions? Okay. Yes, of course, I don't see.
Sorry. From your experience through the hospital work, have you seen which type of germline mutations are mostly predisposing for leukemia over others? I mean, have you seen specific terminating germline mutations, or is it copy number variations over other?
But it's an excellent question. We do know not that much about copy number variations, but it's an interesting question simply because the techniques are not that advanced like in sequencing. Of course, everybody gets sequenced very soon nowadays. So we know much more about specific genes activated or inactivated by point mutations. And they are more or less not specific for B precursor ALL, but there are gene lists. I said Icaros, PAX5, and several others. And with the copy number variations, as I said, it's likely that they do play a role also in connection to leukemia susceptibility, but they are much less well described, I would say.